Hey everyone, I'm Dr. Indira. Today the topic is glycolysis. This video is for everyone who likes to study glycolysis in detail. So let's get started. Have you ever thought what will happen to the food after we eat? Of course, it undergoes the process of digestion and absorption. And the food we eat consists of carbohydrate, protein, fat, vitamin, mineral, fiber, water. Okay, so today we are just going to study about the carbohydrate, or we can say a form of glucose. Okay, form of glucose. So glucose consists of 6 carbon compound, 1 2 3 4 5 and 6. So the word glycolysis itself means glycolysis is derived from the Greek word glycolysis. Glyco means glucose, lysis means breakdown. So the glucose will be broken down equally into a two part. From the 6 carbon compound to from 6 carbon compound, it will convert into a 3 carbon to 3 carbon compound. So during this process, it is going to release an energy from a substrate level phosphorylation, meaning the substrate will release inorganic phosphate that will bind with the adenosine diphosphate that is already present in the cytosol to produce an ATP. So this is an example of substrate level phosphorylation. So glycolysis means it involves the breakdown of glucose to pyruvate while using the free energy released in the process to synthesize ATP from ADP and inorganic phosphate. So this is glycolysis, breakdown of glucose into two equal part, and also producing ATP by substrate level phosphorylation. So the glycolysis consists of 10 reaction sequence and is divided into, some book mentions it is divided into 2 stages, whereas some book mention it is divided into 3 stages. If we are to say the glycolysis is divided into 2 stages, then it consists of energy investment stage, energy recovery or energy generation stage. And next is glycolysis consists of three stages. It consists of 3 stages, that is energy investment phase or priming stage, splitting stage and energy generation stage. Okay, so the complete pathway of glycolysis was elucidated in 1940, and this pathway is also often referred to as Emden-Meyerhof pathway or EM pathway, in honor of the two biochemists who made a major contribution to the knowledge of glycolysis. So they are Sir Emden and Sir Meyerhof. Okay. So let's look on to the silent features of glycolysis. Glycolysis occurs in a cytosol because all the enzymes that are required for glycolysis are present in a cytosol. Glycolysis consists of aerobic and anaerobic. When the glycolysis occurs in the presence of oxygen, then it is called aerobic glycolysis, and its final product is 3 carbon compound pyruvate. So when this aerobic glycolysis is occurring in the glycolysis, the NADS that is produced during the glycolysis can enter into the mitochondria through malate aspartate shuttle and pass through the electron transport chain and participate in oxidative phosphorylation to produce an ATP. So there will be around total of 8 ATP according to old calculation, or 7 ATP according to the new calculation. That is about aerobic glycolysis. So when we are to talk about anaerobic glycolysis is a process by which the glycolysis occurs in the absence of oxygen. So the NADS that is produced during the 7th step of glycolysis sixth step of glycolysis won't be able to enter into the electron transport chain. Okay, so it's not going to produce the ATP that would have been produced from the NADH. So only 4 ATP will be produced, and 2 of the ATP will be utilized to pay the 2 ATP that has been consumed in the initial stage of glycolysis. So it will only have a 2 ATP, okay, during the anaerobic glycolysis, because it is unable to enter into the electron transport chain because there is no oxygen. So glycolysis, when glycolysis is occurring in the cytosol. Okay, so whenever there is a absence of mitochondria in some of the tissue, okay, some tissue lacking mitochondria, then in that case glycolysis provides ATP, for example, erythrocytes, cornea, lens, etc. So glycolysis is the major pathway for ATP synthesis in tissues that lacks mitochondria, for example, erythrocytes, cornea, lens, etc. So let's look on to more silent features of glycolysis. So brain requires a regular supply of energy. So that's why now glycolysis is here to provide a regular supply of energy to the brain. So the intermediates that are produced during the process of glycolysis will participate in other metabolic pathway. So glycolysis, for example, gluconeogenesis, hexose monophosphorus pathway. So there are many more. So glycolysis is a central metabolic pathway with many of its intermediate providing branch point to other pathway. Okay, so glycolysis, glucose conversion of 6 carbon compound glucose, conversion of glucose into 3 carbon compound either pyruvate or 3 carbon compound lactate. So if we are to reverse, because most of the step of glycolysis are reversible, so if we are to move from the end part of glycolysis, then we are able to produce glucose at the end. So to produce glucose, what we need to do, we need to pass through the 3 steps of irreversible steps, 3 steps of glycolysis which are irreversible. So if we reverse the glycolysis, along with alternate arrangement at the irreversible step, that will result in the synthesis of glucose, which is also called gluconeogenesis. Okay, so now we are in the topic glycolysis. So before starting the pathway of glycolysis, we need to know what is the main substrate that is required for glycolysis, and of course it's a glucose. So where do we get this glucose from? Of course, first source of glucose is the food we eat. So after the food we eat, those food will undergo digestion and absorption and enter through the gastrointestinal tract into the blood capillaries, into the blood vessels, and then after that it will enter into the cell. So first source is the food we eat. Second source is glucose that is stored in the organ in the form of glycogen. So liver glucose that is stored in the liver in the form of glycogen, it will be released by the process of glycogenolysis, and that process will provide glucose to the blood vessels. Okay, increase in glucose will be seen in the blood vessel. So liver is the major organ that will contribute to the source of glucose in the bloodstream. So why not other organ? So is glucose not stored in the other organ of our body? No, glucose is stored abundantly in the muscle as well, but the glucose that is stored in the muscle is not able to provide glucose to the bloodstream when required, because of the one fact that is enzyme. Okay, liver consists of the enzyme that is required for glycogen and step of glycogenolysis, whereas that enzyme is absent in the case of other organs such as muscle. So that enzyme is glucose 6 phosphatase, the enzyme that is involved in the release of glucose from glucose 6 phosphate is glucose 6 phosphatase. So this enzyme is absent in the muscle. That's why it is only present in the liver, intestine and kidney. So that's why those organ can only supply the glucose to the blood vessels. So next source of glucose is gluconeogenesis. So gluconeogenesis is a process of formation of glucose through non-carbohydrate precursors. So gluconeogenesis will also supply glucose to the blood vessels. So majorly there are three sources of glucose. One is a dietary, from the food that we eat, and next is from the glycogen that is stored in the liver and by the process of gluconeogenesis. So by these sources we will obtain glucose in the blood stream. After that, glucose will be carried to the cell, from the exterior to the interior of the cell or in the cytosol. Okay, that is how glucose will be carried to the cell, because glycolysis occurs in a cytosol. Glucose needs to be present in the cytosol in order for the glycolysis to occur. So there are major glucose transporter, for example, like there are two major glucose transporter: facilitative bidirectional transporter and sodium dependent unidirectional transporter. When we are to talk about facilitative bidirectional transporter, it consists of glucose transporter 1, 2, 3, 4 and 5. So glucose transporter 1 will help to transfer the glucose through the tissue of brain, kidney, colon, placenta, erythrocyte by the process of glucose uptake. Okay, similarly glucose transporter 2, 3, 4 and 5 have their own specific tissue where it transport the glucose. So glucose transporter 4, it will transport the glucose through the muscle and adipose tissue by insulin stimulated glucose uptake. So this is another different topic. Okay, but today we are focused on only the study of glycolysis, so we will focus on that. Sodium dependent unidirectional transporter. So this sodium dependent unidirectional transporter 1, present in the small tissue of small intestine and kidney, will do an active uptake of glucose against a concentration gradient. So basically these are some of the transporter that are present in our body to transport the glucose inside the cell, inside the cytosol, where the glycolysis can occur. So now we are ready to move on to the glycolysis pathway. Okay, first is energy investment phase. So here the energy will be invested. Similar to when we start a business, we need to do an investment, then we have to work hard, give our dedication, time and energy, then we will get a chance to enjoy the profit. Okay, similarly in the initial steps of glycolysis, first the energy has to be invested. So now glucose has already entered inside the cell. Now glucose will be welcomed by either hexokinase or glucokinase, the enzyme present in the cell. So hexokinase or glucokinase will welcome the glucose by giving a phosphate, and that phosphate is obtained from adenosine triphosphate. So here the ATP has been consumed. So when glucose gets a chance to receive the phosphate, it receives very gladly and keeps that phosphate at its sixth carbon pocket. So that's why it becomes now glucose 6 phosphate, because glucose has phosphate at its sixth position of carbon. So you must be wondering why this one direction arrow. So this one direction arrow indicates the reaction is moving in a one direction, meaning the reaction is unidirectional or the reaction is irreversible. It can't be reversed back. So now there are three irreversible steps of glycolysis. So the first irreversible step of glycolysis is occurring in the first step of glycolysis. Okay, so you will notice that all the enzymes that are involved in the irreversible step of glycolysis, their name are ending with the word kinase. For as you can see here, the name of enzyme is hexokinase or glucokinase. So you must we also wondering why this there is a presence of magnesium. So let me explain you. Since in adenosine there is an attachment of 3 phosphate together at the same time, okay, alpha, beta, gamma position, and this phosphate consists of oxygen which are negatively charged. Now this negatively charged oxygen tend to repel each other, making them very unstable. Okay, so what magnesium does is it will seal the negative charge of either alpha or beta, or seal the negative charge of beta or gamma. So when it seals the negative charge of the oxygen of phosphate, then this phosphate of gamma position will be free, so that it will be accessible to attach with the glucose. So now the glucose can receive the phosphate to become glucose 6 phosphate. Also, you must be wondering why glucose has been phosphorylated. So there is an importance of phosphorylation of glucose. Glucose has finally entered inside the cell. Now glucose is required inside the cell to undergo the process of glycolysis. So to prevent the glucose to pass out from the cell to the outside, to prevent glucose to exit out of the cell, glucose is phosphorylated. Now glucose is negatively charged, which make it impermeable to pass through the cell membrane, which act as a hydrophobic barrier. That's why the glucose has been phosphorylated. Also, you must be noticing I was saying like the two enzymes, hexokinase and glucokinase. So these hexokinase and glucokinase are isomer of each other. Okay, hexokinase and glucokinase are isomer of each other. Actually there are 4 isomer of hexokinase 1, 2, 3 and 4, but hexokinase 1, 2 and 3 are quite similar to each other. So this is hexokinase 4, which is also called a sorry for the spelling, glucokinase. Okay, glucokinase. So let me tell you some of the difference between these two isomer. Hexokinase is present in almost all the tissue, whereas glucokinase is only present in the liver parenchymal cell or in the beta cell of the pancreas. Okay, hexokinase, the product glucose 6 phosphate, it is inhibited by its product glucose 6 phosphate, whereas this glucose 6 phosphate is unable to inhibit the glucokinase. It is not inhibited. Glucokinase is not inhibited by its product glucose 6 phosphate. Okay, so next is the Km value is low and the affinity is high, meaning even in the lower concentration of the glucose, this hexokinase enzyme will act on the glucose. And also hexokinase has a very high tendency to attach to the glucose, whereas in case of glucokinase it has a very high Km and a very low affinity, meaning high Km means there should be a higher concentration of glucose, than only glucokinase will act on the glucose. And also the affinity of glucokinase for glucokinase is less. It doesn't actually want to combine with the glucose. Okay, until and unless there is higher number of glucose, then only it will start attaching to the glucose. So let's look on to some of the other differences of the hexokinase and glucokinase. So hexokinase is not directly induced by the insulin. Okay, it is not directly induced by the insulin, whereas in case of glucokinase it is induced by the insulin. Hexokinase catalyzes the phosphorylation of all the hexoses such as fructose and mannose, whereas glucokinase catalyzes the phosphorylation of only glucose. Okay, So these were some of the difference between the hexokinase and glucokinase. So this was all about the step 1 of glycolysis. Now let's move on to the step 2 of the glycolysis. Now in step 2 we have already obtained glucose 6 phosphate. Now this glucose 6 phosphate will undergo the isomerization reaction. So isomerization reaction is when there is just a change in the some of the components present in a molecule. Okay, so now glucose 6 phosphate will undergo isomerization reaction to form fructose 6 phosphate. So basically it indicates the aldose molecule is being converted into a ketose form of molecule. So during this process, what happens is the carbonyl group that is present here will be shifted to the middle of the molecule. Okay, so this isomerization step actually prepares the molecule for the next crucial phosphorylation step in the pathway. Okay, so this isomerization reaction is moving the carbonyl group to the middle of the molecule, facilitating the subsequent addition of the phosphate group. So ultimately that will really result in the formation of fructose 1,6 bis phosphate, which is then cleaved into a two equal part, that is two part consisting of 3 carbon 2 triose phosphate. Okay, so that is the importance of this step 2 of a glycolysis. The name of enzyme is phosphoglucose isomerase. It is ending with isomerase because it is carrying out a isomerization reaction in glucose 6 phosphate. So the name of enzyme is phosphoglucose isomerase. Now let's look on to the third step of glycolysis. First ATP is consumed in a first step of glycolysis. Now second ATP is consumed in a third step of glycolysis. Now ATP is consumed, the phosphate of this ATP is utilized and placed in a fructose 6 phosphate. So the phosphate that has been received from ATP is placed at the first carbon. Okay, first carbon of fructose 6 phosphate. So now the name of the product is fructose 1,6 bis phosphate. So you must be noticing there the word BIS, not the di. Okay, BIS. In case of ATP we mention adenosine triphosphate or we have adenosine diphosphate, because the phosphate that is present in adenosine are placed together at a point, so that's why they can be named as a diphosphate. But in case of fructose 6 phosphate, the newly added phosphate is present in a different point of a molecule, like in a first carbon and another phosphate in a 6th carbon. So that's why the name of the product is fructose 1,6 bis phosphate, meaning the phosphate are not present together at one point. Phosphate are present, but they are present at a different position. So that's why the name of enzyme the name of product is fructose 1,6 bis phosphate, and the enzyme that is involved is ending with the word kinase. As I had already told you that the name of enzyme ending with kinase are actually involved in the irreversible step of glycolysis. There are 3 irreversible steps of glycolysis. First irreversible step is occurring at a first step of glycolysis involving the enzyme hexokinase or glucokinase, whereas in the second irreversible step of glycolysis is occurring in the third step of glycolysis, and the name of enzyme is phosphofructokinase. Also this enzyme is referred to as a rate limiting enzyme of glycolysis, which will decide the rate of the reaction to move further. Okay, so the committed step or the committed enzyme of glycolysis is considered as the hexokinase, because after we start this step, this will make a commitment that it will however going to complete the glycolysis pathway. Okay, so this is a rate limiting enzyme of glycolysis, phosphofructokinase. So we have obtained fructose 1,6 bis phosphate. So now we are moving so we have already completed the step 3 of a glycolysis. Now let's move on to the step 4 of the glycolysis, fructose 1,6 bis phosphate, that we have obtained from the energy investment phase. We have already completed our energy investment phase. We have received the 2 ATP, which we need to pay off after we earn the profit, after we generate an ATP. Okay, so now in the splitting phase, fructose 1,6 bis phosphate will be splitted into a 3 carbon compound by the help of enzyme fructose 1,6 bis phosphate aldolase. It is splitted into a 3 carbon compound glyceraldehyde 3 phosphate and another 3 carbon compound dihydroxyacetone phosphate. So phosphate is present in both the 3 carbon compound after being splitted, and this phosphate are present in a third carbon in both the 3 carbon compound. So as you can see, the numbering was like 1 2 3 4 5 6, like in a clockwise direction initially. Now when there is a cleavage of this or splitting of this 6 carbon compound, the numbering system has also been changed. So the numbering will be reversed with respect to the original numbering system. Numbering will be start from the point where the molecule has been splitted. So that's why numbering is 1 2 and 3, and also another for another triose compound, numbering will also start from the same point. So 1 2 and 3 is a numbering system. So this is aldehyde, this is in a ketose form. So this glyceraldehyde 3 phosphate and dihydroxyacetone phosphate can be interconverted to each other. So they tend to they try to maintain the equilibrium quantities between both of these. So when there is a decrease in dihydroxyacetone phosphate, then glyceraldehyde 3 phosphate will convert into dihydroxyacetone phosphate and vice versa. And this is done by the help of enzyme triose phosphate 3 carbon compound containing phosphate, triose phosphate isomerase. So isomerization reaction is occurring in a 3 carbon compound containing phosphate. So that's why the name of enzyme is triose phosphate isomerase. So this is it about the glycolysis part 1. So if you have any question, then please mention it in a comment box. If you like how I explain the topic, or if you think it's valuable, then please subscribe the channel, share it with your friends as well, and please you are welcome to give a positive and constructive feedback. See you again in a next topic of glycolysis, that is glycolysis part 2. Thank you for watching the video.